Okay, so I'll do a quick introduction. So thank you all for being here today in the auditorium and online. So today we have Seth Davis, who is a chair of with the chair of plant biology at the University of York in the UK. So Seth obtained his PhD from the University of Wisconsin-Madison, followed by a research fellowship at the University of Warwick in the UK, and moved to the Max Planck Institute for Plant Breeding Research in Germany as a project leader and then research group leader, and habilitated at the University of Bonn in Germany too. In 2013, he returned to the UK as a professor at the University of York. And so there, using Arabidopsis and barley, his group has worked on mechanistic interconnections within the circadian clock. In there is use of system modeling, metabolite profiling, cell biology, quantitative genetics, and molecular genetics to show how allelic variation in clock genes are associated to developmental and metabolic traits associated to crop yield, particularly in response to climate change. So thank you, Seth, for being here today. And yeah, you have about 30 minutes, 45 minutes, as long as you need. Thanks.
Okay, so I have 30 minutes, as I was told. I probably should go a bit quick, I guess, rather than introducing too many things. But I do want to spend some time, because I'm not a computational geneticist, just introducing some of the things that have been interesting to me. And for those of you who plan on using this time to fall asleep, I just want to remind you what my lab is interested in. And that is for all organisms that live on the terrestrial land space, you spend half of your life in the light and half of your life in the dark. If you find yourself at the equator, then it's a 12-12 day year round. And if you find yourself away from the equator towards one of the poles, you still get half of your life in the light and half of your life in the dark. But then there are seasons in that where you get more light or more dark. And that's been my lab's main research interest. It's interesting being at the Roslin because I'm a plant scientist. I have published rarely on animals, and the animals I have published on are things like nematodes. But there you go. Just to highlight what my lab is interested in one slide. So this is kind of 35 years of my career in one slide. And that is light, in addition to providing energy for plants through photosynthesis, is actually an informational signal. So if you grow a pig in the dark, or you grow a pig in the light, you get a pig, okay? If you grow a plant in the light, or you grow a plant in the dark, you get a morphologically distinct organism. So development in plants is dependent on the light environment. So we've been working on a variety of things like the color of light, the direction of light, and this is the thing that we're spending most of our time right now, and that is the duration of light that exists within the day. And I'll just talk about some of those things briefly. So in plants, essentially the same circadian clock that drives our sleep-wake cycles, plants have a circadian clock as well, and it drives not only daily information, should it be doing photosynthesis, should it be protecting itself from cold, so one in the day, one at night, but it's also important for having a seasonal clock. So how do we tell the difference between winter and summer? So if I go outside at 8 p.m. and I look up and it's light, I would conclude it's summer. And if I go outside at 4 p.m. and look up and it's dark, I would conclude it's winter. And that is literally how plants create a seasonal sensors. They have photoreceptors. Maybe something new for you to know is that plants have a visual system just like we animals do. They just see at a different time scale. Rather than looking at a micro and milliseconds time scale, they look at a minutes and hours time scale. They have a variety of photoreceptors. And when you couple that to the circadian clock, then you can determine what season you're in, okay? So the circadian clock of plants controls essentially all metabolism and all physiology and all development. So we use this as our kind of impact case that if we want to understand the circadian clock, it would be relevant to traits that are either ecologically relevant as organisms are being confronted with climate issues and having to move to different photoperiods, or for crop genetics, which is an easier thing to sell to governments, is that you're trying to make barley better. I'm in Scotland, so we want better barley. For those of you who don't know, barley accounts for 10% of the Scottish GDP. So it's second only to the energy industry. So you here in Scotland are exporting headaches around the world. So congrats. Okay. And my lab predominantly, I'm not going to spend a lot of time on molecular genetics. I just want to orient the clock to you. We need a way of asking a plant what time it thinks it is. And what we've done is we've taken rhythmic genes, so controlled by the circadian clock, and have them under the control of the enzyme that causes this animal, the firefly or lightning bug, to light up. And when we make transgenic Arabidopsis plants, a little weed that we have used for model system research, then we get transcription driving this enzyme that makes light. So we can measure transcription simply by photography. So all we have to do is photograph the plant, and we can know whether a gene is being expressed or not expressed. So what we have here are two genes that are expressed in the morning, or in the middle of the day, or in the evening. And these plants were grown for 7 days under 12-hour light, 12-hour dark cycles, and put under constant light, constant temperature. And a photograph was taken every hour, and then a movie was made. And when you do that, you can see that you have these two groups come up first, and then this one, and then this one. So we can measure RNA expression of a single gene in an organism at about a 30-minute time resolution for about a month. Okay, and we can also do that in an individual living organism. So a lot of RNA sequencing experiments are actually population dynamics, right? You kill one pig, you kill another pig, you kill another pig, rather than looking at the expression of a gene product over the life of one living organism. So people, of course, have also co-opted this in mammalian biology as well. Okay, so what can we do? So what my lab and a bunch of other labs did about 25 years ago is mutant screens, where the phenotype was the plant would blink differently than the wild type plants blink. Okay, so that's the phenotype. So this is an example. You have all these plants blinking away, and you can see this plant is blinking differently, right? It's the flashing of light that's coming is different. The organism's still alive, so we can take that plant off of an agar and put it into soil, let it grow up and collect its seeds. So this is a non-destructive process. This is an example of a mutant phenotype from the gene product that's defective in the morning, so time for coffee. The plant can't detect the dark to light transition. And what happened in the original genetic screens, work from Zhao Junding, is that whereas in the wild type you get this rhythm of expression, the mutant has this 2-hour phase shift. So it's very easy for us to see this. Now, of course, no one cares that a plant is blinking differently, right? What we want to know is that there's some kind of developmental or metabolic defect in this organism. So the plant does have whole plant phenotypes. And again, this is an experiment I think almost everyone in the room would have done. So if you don't water your plants, they dry out and die, right? So I think everyone did that one, right? So, and that's what we can see here with Arabidopsis. If you don't water your plants, they dry out and die, okay? But this mutant doesn't. So this mutant is an example that one of the circadian regulated traits in plants is water use efficiency. And this plant is extremely drought tolerant, okay? So, and we've worked quite a lot on that. Okay, so I'm not going to say too much more about this. This is just to orient you that the circadian clock matters to the plant actually doing things outside. But what I'm going to talk to you today about is this issue here. So here we are on the European continent. And if you look at the photoperiod changes that occur over the season compared to the mean ambient temperature, you can see that these gradients do not overlay. And that is what temperature you have relative to what season are not co-varying. That is, temperature perception and photoperiod detection actually should be genetically separable, yet both of them come to the circadian clock to provide seasonal information. So we decided to look at this by looking at natural variation. So what we did is we took two populations where we took an Arabidopsis plant from temperate Europe or West Africa, or another population from the Iberian Peninsula and from Central Asia. And we made recombinant inbred populations from these that harbor promoter luciferase. And then we could measure quantitative trait loci variation in the speed of the clock, dependent on the environment that was used to set the clock. So this is the work of Eleni Boykoglou. So what she did is she either took this population and light dark and trained it at constant temperature, or warm cool and trained it in constant light, and then measured their circadian behavior here. So the plants are actually being measured under the same condition, constant light and constant temperature. And any difference in these plants' behavior is a memory of the environment that set their clock. Okay, so this is an example of two individual rill populations, rill lines, line 47, line 6. So first off, you can see 47 and 6 behave differently. So there's extensive natural variation in the clock and actually at circadian clock genes. The haplotype diversity is rivals that of resistance genes. So they're amongst the most varying genes in a genome in a population, and either under balancing selection or positive selection. Again, consistent with changing the clock is important for an organism to migrate to a new locality. Okay, so here you can see line 6 has a faster clock than line 47. So the peaks come beforehand. And line 6, you can see, doesn't care whether it was photically entrained or thermally entrained. The rhythms are the same. But line 47, you can see that the clock ran faster if it was temperature entrained. And we've looked at hundreds of Arabidopsis lines and actually looked now at a variety of other plants. If a plant cares about how the clock is set, the clock will run faster if it's been set by day-night temperatures. Okay, this was a bit surprising for us because people had always supposed that what sets the clock in plants is the dark to light transition of dawn. And that's actually dogma in the community. And a lot of our work is actually challenging that as to how temperature also sets the clock. Okay, so you do any recombinant inbred population, you get a trait. In this case, the trait is the frequency of the rhythm. So the distance from one peak to another peak is how long it takes the plant to detect a day. And we can measure that as a QTL. So we can measure the QTL for one plant might think a day is 23 hours, another plant might think it's 26 hours. So if I grabbed any one of you here and I put you in a box, your clock might be running at 25 hours and your clock might be running at 23 hours. And she would be the lark and you would be the owl. So you can do these same kinds of QTL measurements in animals to try to figure out the natural variation in mammalian circadian rhythms that exist in populations. So we get QTLs. Usman Anvar and Eleni Boykoglu decided to go at this QTL for a variety of reasons. It's a QTL that has a light-dependent short period, whether it has been entrained by either temperature or light. And because this is a Arabidopsis, we were able to map-based clone the QTL to the nucleotide. So it's one of the nice things about being in a model system is that we could actually see recombination at either SNP on the parental lines in this. Using a variety of technologies. And that point mutation was in an existing clock protein called early flowering 3, L3. This is the high resolution protein structure we have for this. So this is the state of the art. And I don't want to spend too much time about this because it's not a mechanistic talk. But I do want to spend a little bit of time talking about what's L3, just to give you a flavor for the kinds of genes that we work on, the kinds of things that we do. So early flowering 3 loss of function is photoperiod insensitive. So Arabidopsis is a quantitative long day plant. So under inductive, so non-inductive short days, so winter photoperiods, the plant will continue vegetative growth and not make reproductive structures. So you can see this plant has made many, many leaves and does not make any flowers, okay? But under inductive conditions, summer conditions, it will make many less leaves and then will transition to reproductive development. So one of the main things that changing seasons do for plants in temperate Europe is provide information for when the plant should flower in the season. A major trait for farmers, right? So they can predict when they drill seeds, when's the plant going to flower so they know when to harvest the crop. And variation in that is a major trait of breeding. So this mutant, what it thinks is that it's always summer. So even if you grow it in winter, it still thinks it's summer. Okay, so last of this gene product, you can't tell the time of day. As an aside, we do spend a lot of time working on the protein itself. So this is the cellular morphology of the protein. It makes interesting structures. The reason I'm telling you this is to say that your computational genetics group, my lab spends a huge amount of time doing ordinary differential equation modeling. So because the transcriptional gene expression network we work on is an oscillator, we can actually create causality in the arrows that come from ODEs. We know which gene must come before the other gene because it all circles back to each other, okay? So I'm not going to spend a lot of time talking about ODE modeling, but the clock has been a kind of paradigm for how to do this. And we can, for example, make a mutation in one of these gene products and watch what happens to every other gene to see whether the biological data matches the models. And we recursively do that back and forth. So for example, L3 was predicted in our modeling to be a direct transcriptional repressor of PR9, PR7. And if we give too much of these, you can see that we get transcriptional repression and we lose rhythmicity. So this is just an example of a mathematical model predicting an experiment for us to do, okay? And if you care, we spend a lot of time thinking about when proteins are where. So we know this protein is shuttling between the nucleus and cytosol at one time of day. At another time of day, it's only in the nucleus and then makes a protein complex, okay? So again, I know that you guys don't care too much about molecular genetics, but this is just an example of the proteins we work on and the relevance to the computational genetics communities coming back to what this change was that we found in natural populations. It was not a loss of function mutation. It was a single point mutation that affected how the protein works. And what we can then do is look at the gene sequence for this in about 1,000 Arabidopsis lines. And we could try to understand the population genetic structure of this, where you can see within temperate Europeans, they always have one haplotype. But this point mutation, first off, had an identity by descent. So one plant got this allelic variant, but then it was migrated to, like, this is larger than the European continent, okay? And you can see that there are plenty of blue plants where there are pink plants. And what the difference is not a latitudinal or longitudinal association, but rather an altitudinal association. So the prevalence of this haplotype is associated to Arabidopsis plants that finds themselves at the top of mountains rather than in valleys. And that makes actually a lot of sense to us because you have very bright light and the color of light is very different. I mean, for any of you who've been skiing, you know that you can get a sunburn really easily because you have a lot of ultraviolet light. So this is an example of how changes in the environment can lead to selective pressures at clock genes, okay? So we decided to actually test this further, and what we did then is we bred for Arabidopsis populations where the progeny from a cross, we would select for plants that either had a really, really fast clock or a really, really slow clock, and then ask what happens to the plants under normal day-night cycles. So if you have a fast clock, does that mean that the patterns of gene expression get earlier in the day? Because in a day-night cycle, the clock can only be 24 hours. Your 25-hour clock and your 23-hour clock under day-night cycle, it will be reset every day. So what it is that you have phase advances for things like you would wake up early and he would wake up late, right? Those are the kinds of things that you see. So is that what we see in plants? So we made another recombinant inbred population. We crossed two inbred lines to each other. And in the F2 plants, we selected plants that either were really slow or really fast. And then after we made a recombinant inbred population, then we grew the plants outside, right? So how do Arabidopsis plants behave to actual seasonal changes? Because of recombinant inbred lines, we can germinate them in June or July or September and ask, does the outcome of your life depend on the season of your birth, okay? We can also germinate them in little microtiter plates growing in this little greenhouse outside, and then shift them to our light measuring machine so we can actually measure the clock, in addition to having an army of undergraduate students measuring everything you can. How big is the leaf? How green is the plant? How much seed did you make? I mean, whatever you want to measure, okay? So the first thing we can see is that, yes, this is Matt Rubin's work here, is that the behavior of the clock depends on the moment of your birth, right? So this is consistent with what we would have thought of from our light, dark, and warm, cool entrainment experiments. So you can see that whether you have a QTL for the clock behavior depends on the season that you germinated. Okay. As an aside, one of the things, because we had a little weather station out there, what we actually could correlate is that nighttime coolness was what was setting the clock on these plants, not daytime light, which was actually quite interesting to us and something we need to really evaluate. But it also allowed us, because again, your computational genetics community, we do a lot of pathway modeling to understand the statistical relationship of traits. So how fast the clock runs or when the timing of gene expression are do correlate with how many progeny the plants make, which of course is for most crops the main trait. How much seed do you get per hectare, right? That's what breeders breed for, is yield per hectare. But what was really surprising to us is that this went through not only how big the plant was and when did it flower in the season, but went through this trait here, the number of branches each plant makes. So this is what the data look like. So if you have a fast clock growing outside, you make more branches. And if you have a slow clock, you make less branches. And for those of you who aren't plant scientists, this is what less branches and more branches look like. Now, obviously, having more branches means making more flowers, means making more seed, okay? So we don't understand yet why you would want to ever have a slow clock, because you would make less seed, okay? But we certainly see this correlation in the field. Okay, so the final thing I wanted to talk about for our functional data analysis is this idea about, so here we are in York, halfway between Edinburgh and London, is, you guys have thought a lot about this kind of what are the photoperiods that we have, where right now is actually essentially the spring equinox. So in Edinburgh, tomorrow the sun will rise 8 minutes earlier than yesterday. Okay, so jet lag is not something from the space age, it's something selection has acted on, particularly as you come to extreme northern latitudes, such as we find ourselves here. But if you actually look at the difference in the photoperiod changes, you realize this is not a sigmoidal, right? Actually, what you have are rapid changes followed by slow delays. So the rate the photoperiod changes over the season are not constant because of the incline of the Earth along its rotation. And if you look at the temperature that occurs, what you'll see is that the photoperiod changes actually are not circular to the, and we know this, right? Spring is a different temperature than autumn, even though they both are 12-12, right? So again, this is the kind of thing that we're interested in. And what we've done is we've made a recombinant inbred population between an Arabidopsis line from temperate Europe and from equatorial Africa. This is a plant where there are large photoperiod changes, and this is a plant where there's no photoperiod changes. And there are also a lot of other differences in the environments these two plants came from. So particularly the difference in daytime and nighttime temperature and the difference in seasonal temperatures. Okay, so this is what we work on. We work on trigonometric functions, right? So who here can remember their trigonometry? Okay, I'm going to give a trig lesson now, okay? Okay, so with the frequency, the difference between peaks could actually be the frequency measured anywhere. We could measure the frequency from here to there if we wanted to, the inflection, or the phase, the peak of gene expression or amplitude, which is 2 times the difference between the peak and the trough. So when people do circadian measurements, what you're doing is you're creating a time series that's the sum of a variety of Fourier transformations that determines the period. And there's a lot of black box software that clock labs use. So that you put in rhythmic data, you push a button, and it tells you the clock is 27.6, okay? And most clock labs don't know actually what the underlying maths is, and it's this, okay? So, that's fine. But there's a big assumption that comes in this, and that is to be able to actually model rhythmic gene expression data, it makes an assumption that your data are stationary. So stationarity for time series data means that the function is not changing over time, okay? And we've showed that every data set anyone has ever made ever is non-stationary. That is, it is an inappropriate way to model the data, okay? So just to remind you of what this looks like. So this is a trigonometric function. So we're going up the hill, down the hill, under the hill, up the hill, okay? So down the hill, up the hill, right? So this is how you can plot these things. So if the data are stationary, then you do not change velocity. Velocity is constant and acceleration doesn't change. That is, you end up making a circle. If data are non-stationary, then if you were to map velocity by acceleration, you would not get a circle. Okay, so here's some examples of what non-stationarity could be. So the period, the frequency could change over time, something that happens quite frequently. The amount of gene expression could change over time, or could be less over time. This is a very common thing. That is, we might have rapid rises followed by delayed. Okay, so the skewedness is a really common feature of gene expression in mammals and in plants. And also the patterns of expression can change. And we know from ordinary differential equation modeling, you cannot make a rhythmic gene expression if you don't put noise into the system. So this kind of with errors is actually part of the system. It's not measurement error. Genes that are rhythmically expressed need to have error for them to be rhythmically expressed, which leads to stochastic variation of gene expression being a trait itself. Okay, it could be a whole other seminar I could give, but there will be individuals in a population who will be very faithful in their gene expression pattern, and other individuals will have enormous stochastic variation. And we can measure that because we can measure gene expression in individuals rather than populations. Okay, so this is what Sarah Locke has been working on. So this is one plant's expression profile over a time series of about 4 days. And she can think about all of these things, period change, phase changes, and she can measure all of these things, that's fine. But what she's been interested in is, rather than looking at all these patterns here, is to describe it as a function. That is a mathematical equation that explains the data. So we're making a statistical model by having a function that explains the data. And what you end up with is something that's a smooth curve. Now, this is a perfect fit of the data, and this is, she spent a lot of time doing the mathematics for this. This is all black box now, so if you guys want to be customers of ours, all of this is going to go on CRAN, and we're going to have some methods paper about how do you derive functions of time series data so you can explain it in this way. But once we have a function, so x equals y is what we mean by function here, we can now start thinking of this in the terms of calculus. And that is, we can start calculating derivatives. Rather than looking at the periodicity of the frequency or the phase of gene expression, what we can rather look at is the first and second derivative of this, which is the velocity and acceleration. And this is more interesting biochemically, right? People will often look at the phase of this and say, I can find the peak here. But by definition, that's when nothing's changing. What you really want to know is when change is maximum, which is the inflection phase. And you could probably guess it here somewhere. But once you have the function, you can calculate its derivative and know exactly where the velocity inflection phase is. And you can calculate that. And the acceleration. How is the speeding up or slowing down of gene expression over time happening? We can measure that now directly in individual plants. So this is an example of using the method. We've looked at this for a time series of proteomic data. So this is, you grind up an Arabidopsis plant at different times of the day if it's either been grown under summer conditions or winter conditions. This is a particular protein by peptide counts from mass spec. And you can see that the periodicity of this particular protein is the same. And the phase, the peak of its abundance, is also the same. But by your eye, they're not the same, right? So we now have a way of actually calculating the differences in this. That is, we can measure the function that explains these data. And this is what that looks like. We can get a function for protein accumulation, and we can do that for every protein in the proteome. And we can map that as principal components. So in the same way that you're used to thinking about looking at the first and second principal component of data, you can also look at the first and second principal component of the function. So you can do functional principal component analysis. And when you map every protein that exists in a plant, it actually ends up making this donut shape. We weren't actually expecting this, but actually it's really obvious to us what this is. And that is, we don't have to make assumptions of whether a protein is or is not rhythmic. All we're measuring is a protein's accumulation over time and measuring the function that fits that. Mapping that function, what we see is that there are proteins that come up at dawn or midday or evening or midnight or the next day. That is, we can actually calculate when every protein is expressed without making any assumptions of it being a rhythmic protein or not. Okay, so the whole point of this is just to say that we have this method now where we can calculate functions, and it does not matter that the data are rhythmic. So if you just have data that's off and then comes on, but maybe in one organism it comes off earlier or on later, you can still calculate a function to that. Okay. So now we can go back to what we care about, and that is the behavior of our recombinant inbred lines. So we can measure each of the plants, we can calculate a function for all of them, and we can calculate the changes of them as a population. So what we've done is we've grown plants either under winter conditions, transferred to them summer, winter conditions, and transferred them to winter as a control, or summer to summer as a control. And what we're particularly interested in are the changes in gene expression that happen when a plant is experiencing a photoperiod it did not expect. Okay, but of course we will get all this other information as well. So what does this look like? So we're measuring about 200 gene expression points per plant of about 20,000 plants, means that we have about 2 million measurements that we can calculate the function of these. And that whole process takes a couple minutes on a laptop, okay? So this is what all of our data look like together. There's lots of plants going up and down. And this is really the whole point, is that there's actually, it took months for us to work out how to do the functional data analysis. Employing it is not computationally intensive at all. So if you have any time series data, you can feed it to this thing and you get a function that fits your data, okay? And like I said, what we're particularly interested in is how you move from these winter photoperiods to these summer photoperiods. You can see at this moment here, the plant is experiencing an environment that it was not expecting there to be. And you can see that in the individuals. So these are the individuals that did not have the shift, and these are the individuals that did. And you can see a whole variety of features that are different in here. And each of those features are mathematical features that tell us about what's happening to the clock, how is it speeding up and slowing down, but they're also traits that we can measure to understand the quantitative genetic difference that explains how the time series was altered by the shift in the environment. Okay, so this is how we would used to do this. We would have some rhythmic data, and at any given point, we could just take a snapshot and say, what's the QTL at this moment? And you could just sum them all up. But we don't need to do that anymore because our data are not discrete points. They're functions. So we can actually now measure quantitative trait loci of the whole function as a trait. So what does this look like? So this is QTL mapping of composite interval mapping of a biparental mapping population. And you can see that there are some QTLs that are over a threshold. But when I start this movie out, what you can, you can see that there are QTLs, they come and they go. That is, whether a QTL exists is only in the morning, but not in the evening, right? So before, we'd only be having the average QTL of the behavior of this trait over the life of the organism. But now not only do we understand the allelic variation in the individuals, we know when that allelic variation matters relative to the day. And of course, when we do these environment shifts, then we can generate additional information. What was the environmental responsivity of those patterns of expression? And we can also connect this to traits. Again, one of our favorite traits, flowering time. So when plants flower relative to their birth. And you can see here that what we ended up with is the amount of time that the plant flowers is associated to the performance of the time series as a seedling. So by measuring the speed of the circadian clock in the first 7 days of life, we can actually correlate how long the plant will live before it makes seed. Okay, so this was a quite incredible result for us. As another result, I told you we care a lot about stochastic variation. So for genetically identical individuals, may every individual flower the same, or there be a large standard deviation between them, okay? And the standard deviation in the circadian performance correlates with having standard deviation in the flowering time. And the standard deviation does not correlate with the absolute, right? So these are separate traits. The point is we're learning new tools that we can very rapidly be able to, you don't have the whole season to measure when your plant's going to flower. So you can bring that down to 5 days, first 5 days of life, and you can predict who's going to be the winner, right? And of course, this is interesting in all of breeding. You want to use ultimately probably machine learning approaches to be able to bring down those breeding cycles with predicting of gene expression networks. So this is kind of where we are. This is the punchline of this, is that we can measure these time series QTLs that come and go. We get QTLs along the chromosomes. Again, it's Arabidopsis, so we can know the gene and look at the population genetic structure. One of my colleagues right now is doing nanopore genome sequencing of 10,000 Arabidopsis lines. So we're going to learn the large-scale structural variation that exists in species. So that's fine. Okay, so that's kind of where we are. And this just as a reminder that, like, if you guys want to be customers to this approach, we're going to have some methods papers on this if you guys want to play around with it, just to see how does time series data finish. And I think I'm at my 37 minutes, but you want me to stop now? I'll say one more thing. Okay. So, like, who cares, right? I can tell you how a weed grows, right? What we really want to know is how can we improve people's lives, right? We live in the UK and our research is legally required to be strategically relevant to taxpayers, okay? And, King Charles, my king, I'm a unionist, okay? So please don't leave the United Kingdom. So, I think he's doing a lot of great things and thinking about what are the mitigation that we need to think about climate change. And again, come back to barley. So barley is an interesting plant because over the last 10 years in the United Kingdom, what was Northern English barley today is Southern Scottish barley. So breeders are already having to migrate the plant to new latitudes because the plant needs a certain temperature to grow appropriately. But now it's in an incorrect photoperiod to how it was bred, and therefore has less yield, right? So we need to somehow put the clock into breeding to make sure that we can maintain yield, okay? So we can look at a historical example of this, and that is, where did barley come from? So barley is one of the 7 ancient crops that were involved in the civilization of European communities. It came from the Fertile Crescent, so kind of Israel, Jordan, Iraq, Iran. And as it migrated as winter barley into temperate Europe to become spring barley, 2 things happened. One was a loss of the ability to perceive the coldness of winter, and a long delay in flowering that occurred. So Dave Laurie, when he was at the John Innes Center, Matt-based cloned this gene in barley and showed that it's the barley ortholog of a known circadian gene. And it's actually the target of this whole L3 story that I told you about. So by having a lot of this protein, the barley plant doesn't flower. Okay, and what he showed was that it is not a loss of function allele. What it is a single polynucleotide change in this so-called CCT domain. We now know what this protein does. It's a transcription factor, and this point mutation actually is in the DNA binding site. So it's not a loss of function. What it is, the protein has less affinity to DNA, but there's still a protein there, okay? And that actually was quite informative to people to understand the mechanism of this protein. But what was particularly interesting is when me and Maria von Korff then became interested in how did we migrate from breeding barley from temperate Europe to Scandinavia or to Russian climates. So what you need is early flowering in a late flowering background. The plants must flower before winter comes, where you get no seed. So that's how breeding happens in these changes and shifts in latitude. So we just, the main breeding stock in this is called EM8, and me and Maria Matt-based cloned this and showed that it's the barley ortholog of L3. which is the gene I just told you about in Arabidopsis. So this has been a huge convergence of people to start caring about this gene product for breeding. And as an aside, back to the molecular biology, this seasonality, PPDH1, what happens in the L3 is that you have huge super amounts of it in the evening. So you restore the late flowering because you're making high levels of a not very functional protein. If you actually look at this genetically, if you remove this gene product in barley, you don't end up with a spring barley allele anymore. Okay, it just becomes constitutively late flowering, and you end up with an epistatic relationship, which would no longer make it useful for breeding. Okay, so ancient breeders have selected unique alleles that would not necessarily come from mutagenesis breeding. And that's where I am now today. So I was just awarded an industrial fellowship of the Royal Society to try to figure out what is the allelic variation that we have here in Scotland for barley, but I live in England, so we're more interested in bred wheat, which is 25% of all protein that is distributed for human consumption comes from wheat, either directly or indirectly, because we also feed wheat to animals and eat those animals. Okay, so wheat is the most important protein producing plant for humans. So we want to understand how we can breed in climate resilience by looking at allelic variation in these. And I just want to give you an example because wheat is polyploid and the genetics is a nightmare. Okay, so like we have nanopore genomes for wheat, and we can look at structural variation that exists. And this whole seasonality story is not variation in coding region, but actually variation in promoter region. So you change the expression of the gene to create dominant phenotypes rather than point mutations that create recessive phenotypes. So you get your head around that, and it becomes a genetic organism again, right? You just need to start looking at the non-coding regions of the genome that affect the expression of target genes. So for example, a deletion in the D genome of PPDH1, the same gene I explained in barley, leads to, there are 3 copies of every gene, so it's hexaploid. So it does not lead to a change in 2 of the genes, but does lead to a change in the 3rd. Okay, so these things can be measured. We can measure each of the individual homologous transcripts for each gene. So it's all possible now using techniques that exist from high throughput genome sequencing. So I think I'm done, and I just wanted to thank you for your time, and I hope I said enough maths in here to make you guys happy. But what we're interested over, let's say, the next 15 years is understanding how we can exploit natural and induced variation to provide food security for both middle income and affluent countries. That's our main strategic goal. Thank you.
And you can repeat the question for the people in that, that would be great. And I'll just stay here for a question online.
Yes, question.
Okay, this is very naive. Ultimately, clocks, not just circadian rhythms, you know, in us all the time, but especially in embryos. So do you think this can be applied in animal embryos as well?
Yeah, so both in plants and animals, you have rhythms that are also so-called heterochronic rhythms over the linear life of the organism. You see, for example, lateral root formation in plants, but like you mentioned, the formation of new tissues during early embryo development. You know, Sidney Brenner won a Nobel Prize for looking at this cell divides to this cell early or late, right? And you can, in the nematode, map that out. And we actually did this in C. elegans, and we actually showed that C. elegans doesn't have a circadian clock. It has a 16-hour developmental clock, right? So you can measure those things in RNA sequencing experiments. Now, the question, of course, is what is the relevance to this for progression of cancer or other diseases or malformation of earlier development? So I think, you know, we want to understand those things. For plants, plants don't get cancer because cells don't move, so there's no metastasis. But you certainly can get, you know, developmental malformations. That's what orchid breeding is, right? All the weird, wonderful monsters you can create. But how you exploit that in plant genetics depends on the trait. For For animals, I think you're almost always going to be looking at healthcare consequences. But certainly looking at time series data over the early development of, for example, mammals is something that would be very easy to apply this kind of data to.
I think actually probably the application would be looking at changes in metabolism.
Right. So metabolism has rhythms at 2 scales. So RNA polymerase has a 12-minute rhythm that occurs, and somehow that feeds into the circadian clock in ways we don't know. And we have 2 circadian clocks. One is a redox circadian clock, and the other is a transcriptional circadian clock. And we know that if you perturb one, you perturb the other. So metabolism, the Calvin and Krebs cycles are both circadian. I mean, you care probably only about animals, so just the Krebs cycle. But the thing is that all of those molecules have a rhythm in the catalytic event of each of the enzymes and overarching rhythm in the whole process itself, right? So if you just, people have done this mostly with mice, but if you just grind up mice at different times of day, you can measure when is glucose 6-phosphate most and less, and you can do that in the liver, heart, kidney, whatever, and you can end up with metabolic time series over all of those organs over circadian and developmental timescales. Also overlaying things like drug addiction or overeating or all kinds of things people have done to the mouse in that. In the plant, we've done less of that. Did I answer your question?
Yeah.
Yeah. No, but I think what's interesting is actually how do time series at different time scales intersect with each other, right? And that's something I think we need to, as biologists, understand more about.
There is no question online, and I don't know what time it is. But... You have a full.
Quick question. Very nice talk, by the way. Really enjoyed it. When you're talking about looking at the phenotypic variation being driven by these rhythms, have you looked formally at how much of that variation in particular settings in animal?
Okay, so the question was, have we looked at how phenotypic variation is driven by, I guess, clock gene haplotype variation? Is that kind of what you're asking? Other labs have started looking at rhythms in perennial plants so that you can look at an individual plant over multiple different seasons. What we don't know we can measure haplotype diversity in clock genes. What we don't know is what were the traits that selected for that haplotype variation. Certainly the speed or functionality of the clock itself is not under selection. It's the traits that the clock controls: flowering time, glucose 6-phosphate levels, light harvesting, freezing tolerance. So which of those things is most important that's selecting for these allelic variations? We don't have the answer to that. We can recursively say that if you have this allelic variation, this is the phenotypic consequence, but we don't know that that was necessarily what was selected for. If that makes sense, I don't know. But, it's always very difficult to say whether a given haplotype is adaptive versus happened and you live with it, right? And I think a lot of molecular ecologists really struggle with that question, actually.
And most times something that authors or seeds, but and in potato, for example, there is this negative correlation between maturity and yield. So earliness, even though it would be good, but it yields less and not very good as soil. Anything about these rhythms influencing this type of negative correlation?
Okay, so how so the question is for vegetative crops that are not the flowers are the harvesting trait, how does rhythms affect the yield performance? Giving potato as an example. So potato tuberization is, so the particular nightshade family is a so-called short day plant. So everything I said for Arabidopsis is the opposite. The inductive conditions are winter and the non-inductive is summer. And Salome Pratt's lab, first she was in Madrid, now she's in Barcelona, has showed that the same pathway that exists in Arabidopsis that's involved in providing information from the leaf to the meristem, which is the stem cell set of plants, to cause the cells to stop making vegetative tissue and start making reproductive tissue, is the same pathway in the roots to determine whether the plants would stop making stems or start making tubers. And it's all of the same components. And all of the clock mutants that I talked about, if you pull the plant out of the ground, you'll actually see large developmental root phenotypes associated to largely clock control of temperature. So one of the mechanistic papers I published a couple of years ago is that the leaf actually provides temperature information to the root in a clock-dependent fashion by having a clock protein move from the leaf to the root to determine branching of the root, okay? And in the nightshade family, we know that the domestication of the nightshades was a similar story to barley. That is, it was sequential introduction of circadian mutations allowed the plant to move away from the origin around the equator, so tropical climates, to more, but also to grow in more arid and brighter light conditions. More of that's known in tomato than potato. But there are a lot of varieties of potato that here in Western countries we don't have that can be photoperiod sensitive or insensitive. That is, they can be perennially tuberizing or not. And that absolutely correlates with allelic variation in seasonality genes, not so much clock genes, but certainly genes involved in seasonality. But coming back to this question, we don't know what the cause of that is yet, right? So you can show that if you have this allele, you make more tubers, bigger tubers, but we don't understand how that allelic variation caused that consequence phenotypically. But certainly the other interesting thing about the nightshades is that they're both long day and short day versions. So it's actually an interesting model species to understand how you would switch. How do you stop being a long day plant, which is the ancestral state, and start being a short day plant? Because about half of the plants growing around the world are short day, including most of our protein crops, the legumes, and rice, which we know nothing about them. So these are important questions. So please, funding agencies, give me more money so I can work on them. But I mean, I think it just shows that how basic science could very, very rapidly lead to improvements in yield per hectare. The only thing breeders care about, yield per hectare, that's it. Is that an answer to your question? Okay.
So there's still no question online. If you don't have a question, a last question in the room, then thank Seth for his talk and for coming today to Scotland.